Hi everyone, it's Dr. LeBlanc. This is a preview of transcription. The central dogma states that DNA makes RNA makes protein. In the last unit, we studied the process by which DNA replicates itself. This unit is concerned with producing RNA from a DNA template. Down below here, you can see a diagram of RNA polymerase that has just copied this stretch of DNA here into RNA. Right here is the start site of transcription, and here is the stop site of transcription. And this RNA polymerase has completed this transcript and is about to terminate when it hits this nucleotide sequence right at the end of the gene. So we're going to be concerned with the process of transcription, which is making RNA. Here is a little GIF showing an RNA polymerase molecule that's just bound to a promoter and is now synthesizing a molecule of RNA. This RNA could be a messenger RNA, it could be a tRNA, it could be a ribosomal RNA, it could be a microRNA. There are many, many different species of RNA in the cell. The trick for RNA polymerase is to find a gene to transcribe. Think of the whole genome. In humans, it's billions of base pairs long. How does RNA polymerase know where to bind and where to start transcription? Well, the process of regulating gene transcription is the topic of another unit, but we're going to be concerned with the actual mechanics of producing an RNA molecule from DNA. In other words, the initiation, the elongation, and termination. Recently, in the journal Cell, was an article called The Structural Mechanism for Rifampicin Inhibition of Bacterial RNA Polymerase. So once again, we see a molecule that's involved in replicating or transcribing DNA as the target of an antibiotic. This antibiotic binds right here in this molecule of RNA polymerase. This is the structure of the antibiotic. And this inhibitor acts by directly blocking the path of the elongating RNA when the transcript is only 2 or 3 nucleotides in length. So very early on in the process of transcription, rifampicin will bind to a RNA polymerase molecule and block the exit of the new transcript. And if that exit is blocked, no more RNA can be transcribed. And if you can't transcribe RNA, you're not going to be a viable bacteria. Another big question is getting genes activated or reactivated in tissue where they're not normally active. There was an article recently in Science magazine called Gene Activation Therapy Presents Liver Damage in Mice. Here's a molecule of DNA, and this green diagram and pink diagram are two ribbon diagrams of molecules of protein that bind to DNA. They're binding in the major groove right here, and on the other side of the DNA molecule on the other major groove. They are called transcription factors. So researchers have found a way to deliver gene-activating molecules, these transcription factors, into specific tissues of a living animal for the first time, turning on genes in specific tissues rather than giving them systemically and turning that gene on in all the tissues. Whole transcription factors like these are a little studied approach to therapeutically changing gene expression. But researchers have now found a way to target specific tissues and get these transcription factors in and turn some genes on that might have been dormant. RNA is a diverse collection of molecules. RNA can be found in a variety of shapes and sizes, messenger RNA, transfer RNA, a lot of different types of ribozymes, in other words, RNA molecules that act as enzymes. And RNA is often bound with proteins in a class of molecules called ribonucleoproteins. And if you go to this website at the Howard Hughes Medical Institute, you can find lots of different types of RNA discussed. So this is an interactive website. I'm going to go through these quickly. Messenger RNA, which encodes the instructions to make proteins. Every 3 nucleotides encodes a different amino acid. Double-stranded RNAs are important in gene regulation. Transfer RNAs bring amino acids into the ribosomes in the construction of new proteins. Ribozymes, many different types of ribozymes. Introns that self-splice, hammerhead ribozymes, parts of viruses. The Delta virus has hepatitis Delta virus has one of these hammerhead ribozymes. Another ribozyme. RNA nuclease P is another ribozyme. And the ribosome, the big factory inside every cell, many of them inside every cell that produce proteins, is actually a ribozyme. The catalytic portion of the ribosome is comprised of RNA. And these are some interesting lectures you can look at if you want. Thank you.